topic will be on our newest lab service, DeepImmu, AI-driven mass spectrometry-based immunopeptidome discovery service. So our topic will be divided into three parts. In the first part, we will briefly talk about what is immunopeptidomics and its significance. In the second part, we will give you an overview of our DeepImmu service workflow. In the third part, we will present you with the demo project. So let's go into the first part. What are immunopeptides? So immunopeptides are MHC-bound antigens that are presented to immune system. There are two types of MHC molecules, MHC1 and MHC2. MHC1 is mainly responsible for presentation of endogenous antigens, while MHC2 is responsible for presentation of exogenous antigens. In MHC1 pathway, endogenous antigens, for example, misfolded protein, aberrantly expressed proteins will be digested by proteosomes into peptides, and these peptides will be transported into ER. And ER, these peptides will form complex with MHC molecules, and if the complex is stable, they will be transported into cell surface for interaction with CD8 T cells. MHC2 pathway is a similar way where the exogenous antigens will be digested into peptides by endosomes, and they form complex with MHC2 complex molecules and send it to the cell surface. Within MHC1 peptide presentation pathway, endogenous antigens can be generated in several distinct ways. So this leads to a topic of non-canonical peptides. So endogenous antigens could be formed by alternation of genomics and or epigenomics, alternation of transcriptomics, also in translational level, for example, translation of non-coding region, insertion of jumping genes, etc. All can contribute to forming of non-canonical peptides. Some of these non-canonical peptides could be unhealthy cell or system specific, and revealing of these non-canonical peptides become a big deal because before this, researchers were looking intensively on the canonical antigens and targeting canonical antigens that are overexpressed in unhealthy system, and targeting them often cause on-target off-tumor effects on patients. So immunopeptidomics research became hot topic since 2016, where one research group identified non-canonical peptide from non-coding region using mass spectrometry-based identification methods. So this identification, surged the research interest in immunopeptidomics from basic research on binding motif identification in preclinical research on target discovery and to clinical research on vaccine design, contributing to the research areas such as cancer immunotherapy, autoimmune diseases, transplantation immunology, and infection disease immunology. Among all these mentioned research areas, non-canonical peptides became hottest topic in cancer immunotherapy. And the neoantigens cancer vaccines has already shown it is efficacy in preclinical research and proceeded into various clinical trials. So, however, there are still challenges in immunopeptidome discovery. Detecting low abundance of peptides and distinguishing between similar peptides with high sensitivity and specificity is challenging. To address this challenge, improvement in mass spectrometry sensitivity and data analysis algorithm are needed. And to identify non-canonical peptides, DeepNovo sequencing with optimized algorithm is also needed, and how to validate these peptides is also a challenge. Keeping these challenges in mind, let's look at how we address these challenges with our DeepImmu workflow. So DeepImmu workflow is what we provide as a lab service here in BSI. And this is the whole workflow overview. Samples are prepared using our in-house developed Neo discovery kit with affinity purification of immunopeptides. And LC-MS data acquisition is performed with our cutting-edge mass spectrometry instruments. And raw data is analyzed using our Pix11 DeepNovo peptidome workflow, which specifically designed for immunopeptidome discovery. We will also perform an immunogenicity prediction and optional cell validation upon customer's request here. Then the report will be generated. So let's look at each step in details. First step for sample preparation is immunoprecipitation of MHC complex. QC Western blot will be performed for checking successful enrichment of complex. Then the peptides will be purified by step elution. After purification, peptide sample will be loaded to mass spectrometry for data acquisition. After data acquisition, raw data files will be analyzed using our own Pix software DeepNovo workflow for overall quality control assessment on both MS1 dataset and MS2 dataset, as well as the data quality between the technical replicates. And measured retention time of peptides will be plotted against the predicted retention time of the peptides to check the correlation between the measured peptides and the predicted peptides as another step for data quality control. So if the quality control passes, we will then go into in-depth analysis of sample data with Pix11 DeepNovo peptidome workflow. So this whole workflow is specialized for peptidome data that combines database search, de novo sequencing, and identification of mutated peptides. We trained our deep learning models using peptidomics data to improve the sensitivity of peptide identification. So, and also in the second round of search, all the database search peptides and the homolog peptides, DeepNovo peptides are combined as a new database, and a new FTR controls will be set to a better estimation of FTR. And the final results will be exported for binding affinity and immunogenicity prediction. Please note that in this whole workflow, if you have NGS data, it's compatible with our whole workflow. So for immunogenicity prediction, so identified peptides then goes into our DeepSelf AI-based personalized model for immunogenicity prediction. This model is developed by learning how our T-cell central tolerance system works. So immature T-cells usually goes through both positive and negative selection to eventually become mature CD8 T-cells. During positive selection, they will be presented with non-self antigens. If they can recognize these antigens, they will go into next step. If they cannot, this will be called as a failure, and then they will go through apoptosis. In the negative selection, they will present it with a self-peptide. If they do recognize this self-peptide, this will be categorized as a failure, and then they will go through apoptosis. If they do not recognize self-peptide, this will be called as a success, and then these T cells will promote it as a mature CD8 T cells. So in our model, we have similar positive and negative selection training process. In negative selection, what we do is our model will be presented for with immunopeptides that are identified as database search or self-peptide from your own sample. And in positive selection, our model will be presented with allele matched IEDB epitopes that are immunogenic. After training, DeepNovo peptides and homolog peptides identified in your samples will be loaded to our model for immunogenicity ranking. So we also did comparison with other immunogenicity tools that are existent. So we did a performance evaluation of 4 immunogenicity prediction tools on 18 cancer patients. Please note that we have a published paper, preprint published paper listed as a reference in our website. If you're interested in details, you can check out our DeepImmu website. So yeah, so for this evaluation, this plot is a performance matrix used to evaluate the accuracy of binary classification model. Here, x-axis is false positive rate and the y-axis is the true positive rate. AUC means the area under the curve. So usually the AUC value ranges from 0 to 1, where AUC 1 indicates a perfect discrimination between the two classes. So higher AUC means better discrimination power over the model. So you can see here, our DeepSelf model has 0.69 value of AUC comparing to all the other 3 immunogenicity prediction tools. We have a higher score on AUC. So after prediction, we have optional in-cell immunogenicity validation experiments done in a live spot system. So upon discussion with customers, target of interest peptides will be synthesized and goes into cell validation. Okay, let's go into the third part, demo service project. So in this project, human lymphoma cell pellets were given to us. Samples were processed by immunoprecipitation, and QC Western blot results shown here. HLA complex were well enriched. After data acquisition and data analysis, in summary, what we got is 11,000 total peptides, in which 10,000 peptides were identified by database search, and 150 peptides were identified as homolog peptides, which is mutated peptides, and 1,300 peptides were identified as DeepNovo peptides. And identified number between each technical replicates were consistent. Let's go into the software details. Like I mentioned in the previous slides, after sample purification, we go through overall QC steps to make sure our mass spec data is good. For this sample, MS1 and MS2 data both in small error tolerance range, and between 2 replicates data quality is consistent. Peptides below 1% FDR range were collected and measured RT time and predicted RT times of the peptides. Correlation were checked as another step of quality check. Then each filtered peptide then is listed with the detailed information. CAA value is here means confident amino acid percentage, which is calculated according to both signal intensity of fragment ions supporting that specific amino acid and the existence of both B ion and Y ion supporting that amino acid. And mirrored plots is shown here comparing the measured spectrum of this DeepNovo peptide and the predicted spectrum of this peptide for further sequencing accuracy assurance. And each fragment ion, master charge ratio, and intensity are listed for further assurance. Then after that, all filtered peptide then exported for length distribution and binding prediction analysis. MHC1 peptides are usually in 8 to 12 amino acids long, and within this range, 9 mers are usually the predominant population. So by doing this length distribution analysis, we confirmed that identified peptide length exhibits typical MHC1 peptide length attributes. And for further validation, binding of prediction is also analyzed, and you can see here over 80% of identified peptides were predicted to be binders. And also binding specificity towards each allelose are also analyzed and shown in a heat map format, suggesting a binding overlap between B1510 and B391 allele. And we also perform an offset plot between the technical replicates, and you can see here comparing the peptide composition between 1 and 2 replicates. They have a big overlap with each other, suggesting a good data quality. We also do binding motif deconvolution of eluted peptides mesh, and you can see binding motif deconvolution of eluted peptides were matched to what was expected, further confirming a good quality of identified peptide or eluted peptides. So after that, after the validation, these peptides will then further exported for immunogenicity prediction in our DeepSelf model, and a list of predicted immunogenic peptides will be provided with detailed information on each peptide, and optional in-cell validation experiments will be discussed with the customers. So, in summary, the challenges in immunopeptidome discovery I mentioned before are all addressed in our workflow, in which low sensitivity and low reproducibility issue were resolved by our high efficiency and reproducibility of immunopeptide purification methods, high quality mass spectrometry data acquisition, and stringent quality control tests. And the challenge of identification of non-canonical peptides are solved in our workflow by DeepNovo algorithm-based sequencing for identifying neoantigens, specifically designing for immunopeptides profiling. And challenge of validation of identified peptides also solved in our workflow by DeepSelf immunogenicity prediction and optional in-cell validation. So if you're interested in our service, these are what we require to process your samples and what we provide the qualitative results that I shown in the demo project, and also a comprehensive report and comprehensive report suited to your own customized research. So that will be all from me today. Thanks for joining us again to this seminar. Yeah.